So thank you very much for the honour of presenting at this sad but celebratory symposium for a great life well lived by this wonderful woman. So I first met Athina, I think in a meeting in Montreal when she was still a graduate student in, I think, 1988, and I learned subsequently the connection with Mark, and we've stayed very, very good friends ever since, including the visit to Cambridge, as Barry outlined earlier, and continuing in other domains besides science. And some of my last memories of Athina is when she came over to England to see a performance by ballet heroine, Sophie Gwilym, with Mark last year, and my own wife, Barbara Sahakian, who's also very friendly with Athina and Mark, and I were really delighted to accompany them to that. Okay, so problems in translation, which of course Athina and Mark have really been more aware of perhaps than anyone else, and in particular focusing on this poverty and naivety in the use of animal models, particularly perhaps in pharmaceutical industry, and the lack of back translational feedback by which one validates such models. And I'm very much motivated myself by this phrase of Mark's, which I think there's a strong element of Athina there, Mark, as well, I imagine, when he said that the drug companies can't even predict the outcome of phase 3 studies from phase 2 studies, let alone from animal models, which they criticise so readily. And of course it's this seamless integration of animal models with the other phases of drug discovery which is so important, and which was so well recognised by Athina. So one of the advantages perhaps we have in formulating animal models in psychiatry, which is so difficult because of the lack of our understanding of the molecular pathology and the pathophysiology of many of these conditions, is the fact that there are behavioural homologies across species which we've capitalised on in the so-called CANTAB battery for measuring aspects of cognition, for example, working memory, cognitive flexibility, attention, and also response control, which is going to be the main subject of my talk. So I've been interested, like Athina, in a transdiagnostic interest in anhedonia, from drug addiction to depression, in the concept of impulsivity, as exemplified, obviously, prototypically in ADHD, and the realization that here we do have drugs that work in ADHD, some of the most dramatic effect sizes in the whole of the area, but we still don't really quite understand how they work at a systems level. We understand that methylphenidate blocks the transporters for catecholamines, atomoxetine, the selective noradrenaline reuptake inhibitor for noradrenaline, but we need to understand more about how these drugs actually work in order to gain ideas for better drugs for treating impulsivity and associated disorders. One of the test paradigms that's commonly used to measure ADHD in the laboratory, as it were, is the STOP task, invented by Gordon Logan. And in this paradigm, subjects, humans, are supposed, they're trained essentially to respond as quickly as possible to one of two options, left or right, as directed by the imperative arrow coming up on the screen. And on a proportion of these trials, however, they have to restrain or cancel that response when a beep is sounded. This occurs on 25% of trials, offset from the imperative signal, and making it very difficult once the response has been initiated to actually stop it. And so impulsivity in this case is measured by slow stop reaction times, which can be computed from the response time distributions. And as you see in some of these studies of ours, both Ritalin and atomoxetine work in adult ADHD to improve response inhibition. So here's the effect of Ritalin, and here's the effect of atomoxetine. So this is good. We have a good human model, as it were, of this clinical efficacy. And then jumping on ahead, we obviously need to understand the neural basis of this, and one of the lines of investigation that we've used has been to study impulsivity as it occurs in adolescence in this very large population in a pre-Brexit study of 10 centres in Europe of over nearly 2,000 14-year-olds eventually. But in this interim analysis, we used fMRI to identify those response systems which contribute to successful stopping. And one can use principal component analysis for this and identify 7 orthogonal systems, here's the eyes of course, but which include the orbitofrontal cortex, the medial frontal cortex, this very important cluster, the inferior frontal cortex on the right side and the cingulate, the basal ganglia, parietal cortex, and the supplementary motor area where we believe inhibition is delivered finally. And intriguingly, first of all, those adolescents who have begun to experiment with drugs, notably nicotine and alcohol, but also cocaine in some instances, had reduced activations in these regions of the orbitofrontal cortex, and in the case of stimulant drugs, changes in the inferior frontal cortex, which predicted the degree of their drug abuse even at this incipient stage, if you like. But intriguingly, there was some overlap also with ADHD symptoms, not complete, but nevertheless, those reporting ADHD symptoms had reduced activity in a bilateral, this frontal error monitoring network, including the anterior cingulate, insular, and right inferior frontal cortex, on failed trials only. So when they failed to stop, this is when this was picked up by these measures. So drug use and ADHD symptoms were related to different networks, but related networks underlying impulse control with some obvious overlap. And also intriguingly, we're able to find that there was a polymorphism which also predicted the ability of subjects to stop, and polymorphism of the noradrenaline transporter, particularly in this region of the right inferior frontal cortex. And the reason that's interesting is because in our studies of atomoxetine, this selective noradrenaline reuptake inhibitor, or largely selective, we found even in normal volunteers, atomoxetine improved stopping compared to the SSRI citalopram and placebo. And in the fMRI study, it actually, on successful trials, it dose-dependently, and this is related to plasma levels of atomoxetine, enhanced the activation in this right inferior frontal locus. So with a combination of pharmacological and genetic and imaging and behavioural evidence, we're able to postulate that one of the ways in which atomoxetine works is by modulating the activity of a right inferior frontal network, which probably integrates activity with the basal ganglia and the supplementary motor cortex. Okay, well, serotonergic manipulations, much less, in fact, ineffective essentially, as would have been predicted by the lack of effective citalopram, which had major effects on the ability to respond to negative feedback in other behavioural tests. And even when polymorphisms of the serotonin transporter promoter were taken into account, there were no differences in inhibitory control between subjects subjected to tryptophan depletion, which is a way of impairing serotonin transmission, and a balanced diet in these human volunteers. We're able to model these effects in rats. So with Dawn Eagle, we designed a task in rats which captures the stop signal task, and in this task, rats are trained to respond as quickly as they can between 2 levers, which leads to the delivery of food pellets, except on trials where a tone occurs between, immediately after or some delay after their first response. And on those inhibition trials, they have to go directly to collect their food pellet and cancel the second response. And using this procedure, we're able to show striking homologies, if you like, with the human data, so that atomoxetine dose-dependently improves stopping in these rats without affecting other aspects of performance. And moreover, serotonin manipulations, which would be predicted to have no effect, even though global 5-HT depletion has major effects on some aspects of behavioural inhibition, not on the stop signal task. and nor indeed does citalopram given acutely much affect performance on this task. So again, some comparability psychopharmacologically between these drug effects in these animal and human paradigms for measuring impulsivity. And Andrea Barry in our lab then tried to assess the central substrates of these effects of atomoxetine by infusing the drug directly into cortex and combining this with pharmacological manipulations to determine the fact that it was working via noradrenergic mechanisms. And as you see in this slide, the biggest effects were shown when the drug was infused into the lateral orbitofrontal cortex of the rat, which may be possibly a homologue of the inferior frontal cortex in humans. And you can see this huge improvement produced by this effect, which was abolished by noradrenergic treatments. So here we have an animal model, back translated as it were, to fully understand it and to identify a noradrenergic mechanism influencing behavioural control of a task which is impaired in ADHD and also in drug addiction, because in our laboratory, Karen Urscher has shown that not only do stimulant abusers have problems with the stop task, so too do their relatives who do not abuse stimulant drugs, suggesting that this may be an endophenotype for stimulant drug abuse, including potentially also nicotine abuse. And so with this in mind, we tried to translate the effects of atomoxetine in these ADHD models to the drug addiction arena, and this is work of another sad loss to neuroscience, Dinah Ekonwadu, working with Barry Everett and Jeff Daly and I, and using this second order schedule that Barry referred to earlier, this very sensitive procedure in the first interval, showing a dose-dependent reduction of drug seeking in the first interval produced by atomoxetine, both for cocaine and actually, intriguingly, also for heroin. Not after the drug was taken, but only in this drug-seeking phase. And the effects are not motoric because animals will still work for food. So we're currently endeavouring to translate this effect to human work, and with Karen Urscher, using these tasks and others, we're trying to show in those cocaine abusers seeking treatment whether there are any effects. And this is preliminary data really, looking at the effects on a drug craving model mentioned before, where subjects are supposed to ignore drug-related stimuli as they count these lines on the screen. It's a sustained attention task. And you can see interesting preliminary evidence that maybe atomoxetine improves the ability to remove the interference posed by these drug-related measures, specifically in the stimulant abusing population. So finally, of course, who would we invite to contribute to a book on translational neuropsychopharmacology than Athina Markou? And she produced a wonderful chapter with Diego Pizzagalli and also De Akavian, a beautiful chapter summarising some of the evidence you've just heard. And I'm so happy to say that we were able to include in the volume a dedication to Athina's enormous contribution to translational neuropsychopharmacology, which Athina was aware of before her sad demise. So I'd like to thank, finally, all my collaborators in these efforts in translation. Thank you very much.